Okay, so I'm going to do a little introduction for apoptosis. We've been talking about apoptosis or programmed cell death all throughout this experimental module. However, I'm going to go a little bit more in depth right now into what it is and how important it is, not only for treatment of cancer through these drug treatments, but also in just everyday cell well-being and body well-being. So we are going to be talking about that. Okay, so apoptosis, or what we call programmed cell death, is a normal physiological response. It happens in our bodies even during the process of embryogenesis, when we have webbed fingers and toes when they are first created, and then through apoptosis all the cells and the webbing die out and you get your individual digits. Similarly, cells in our body, as they grow old, as they get damaged, or they get, whether it's through genotoxic damage or because we've taken something, maybe gotten exposure to some chemical, those cells, as they get damaged or get old, they have a response built in that triggers apoptosis or programmed cell death, so that those cells can die out and other cells can take their place and continue the work that they were supposed to be doing. It is a form of cell suicide, essentially. Now, this happens, like I said, in the growth of embryo, through, and in other normal processes as well. It can also happen as a way for our tissues to maintain homeostasis, to maintain enough cell count to do the work that they need to do efficiently. It is also a reason for a lot of diseases. So it can induce neurodegenerative diseases. Uncontrolled apoptosis of neurons, for example, can cause neurodegenerative diseases. It is also one of the reasons for insulin-dependent diabetes, where the islet cells start to die out through apoptosis as they become damaged or unable to do the work, leading to diabetes. It is a direct effect that you see in myocardial infarction or heart attack, where the affected heart tissue dies off through rapid apoptosis. It can also be in response to chronic viral diseases as well. Infection from viruses can cause damage to organ systems that they are infecting in a way to induce apoptosis as well. The pathway for apoptosis depends upon what it is that is activating it. So there are a bunch of triggers that can trigger the process of apoptosis. This could be a growth factor deprivation or starvation response. It could be because of hypoxia or low oxygen tension. It could be because suddenly they don't have the proper places to attach a substrate to attach to, like loss of adhesion molecules. For example, if we left the cells in trypsin too long and the cells lost all those adhesion proteins, and then we plate them in without trypsin but without serum, so they were unable to replicate and or produce those other proteins, it could also cause apoptosis in that case as well. There are death receptors on the surfaces of our cell. If those receptors get activated through binding of a ligand of some sort, that can also induce apoptosis. And then obviously in the case of radiation damage or chemotherapy treatment, where it is causing genotoxic damage of some sort or cell damage of some sort, it can also induce apoptosis. These triggers work through modulators. Depending upon which trigger you're looking at, it's going to affect a different modulator and activate a different modulator or set of proteins. I have listed a set of example proteins that could trigger these triggers can modulate, and some of them are going to be inside the cell, some of them are going to be on the cell surface. But these modulators are then going to go ahead and activate effector proteins. The effector proteins in apoptotic pathway are caspases. There are many different types of caspases that can be affected, that can be activated in response to these modulators. Different caspases will get affected and activated in response to different modulator. So you can look at which one is activated to kind of guess which modulator might have been activated and which trigger might have been responsible. So once the caspases are activated, they work on their substrates. They are enzymes. They work on substrates like cellular proteins and DNA to chew them up and destroy them and cause the death response. So caspase is a single chain of proenzyme. They are always present inside the cells, ready to go. It's like a reserve army, always ready to go do their work. However, they will only work and do what they need to do when activated. They get activated by the modulating proteins. They contain an N-terminal domain, and they have a small and a large subunit and look very similar to a ribosome in some cases. You can have an apoptotic stimulus activate a caspase. That activated caspase will cause cleavage of its substrates and will release other enzymes and other proteins and activate them as well. So there are three main types of caspases. There are inflammatory caspases 1, 4, and 5 that do not have to cause apoptosis, but can just be an intermediate response to damage happening to the cell in response to UV radiation or chemotherapy. You have initiator caspases that are 2, 8, 9, and 10. They are the in-between between the modulator and effector caspases. They have extremely long N-terminal domains. And when activated, their job is to activate effector caspase. Effector caspases, there are three of them, 3, 6, and 7. They have little to no N-terminal domain, and they are the ones responsible to initiate the actual cell death. So they're the ones that are going to chew up the DNA and the protein and destroy the cell and cause the blebbing and everything else. The one that is going to be always activated as a final response is caspase 3, regardless of which way you activate this pathway. There are two main pathways that caspase can be activated. There is an intrinsic response pathway and there's an extrinsic response pathway. An intrinsic pathway, caspases are activated through internal signaling pathway. So some kind of a damage to the cell activates the mitochondria to release cytochrome C. Usually it is due to p53 activation, which is a tumor suppressor protein. The p53 protein, when it activates, causes upregulation and downregulation of proteins related to mitochondrial function. And these proteins, when they are dysregulated, will cause the release of cytochrome C from mitochondria. That cytochrome C interacts with APAF1, which is an intermediate pathway protein, to create an apoptosome that also includes caspase 9. Caspase 9, activated caspase 9 causes the center of this apoptosome. This apoptosome will then go ahead and cleave caspase 3, 6, and 7 to activate them, which would in turn cause the death response. The other pathway that you can have is the extrinsic pathway. In the extrinsic pathway, there is some kind of a trigger that binds to the death receptor on the surface of the cell membrane. These death receptors, when bound by the pro-apoptotic ligand, will get activated, and they will activate caspase 8 protein. Caspase 8 protein, when activated, will then interact with caspase 3, 6, and 7 to activate those and cause an apoptotic response through that pathway. This is a super fast death pathway or caspase pathway compared to the other one, which can take a longer time and a slower response. When you look at these cells in culture, you can tell the difference between a normal cell and a cell that is undergoing apoptosis because of their characteristic look of, they become more 3D. They start to lose the markers inside, like the nucleus and the other, organelles that are present there. You'll see this kind of blebbing or bubbling on the cell surface. in the early stages. Later on, you will see the remainder of the cell body in the center, and you would see these kind of star-shaped spikes on the sides of them. And finally, you will be left behind with a small remnant of the cell body, almost like a blister, very small cell body with nothing inside, and the cell would have died away. So the caspase activity, the assay that we did, looked at caspase 3 activity specifically. So in this case, we had the active caspase 3 in the protein lysates that you had from your individual treatment. Those served as our source of caspase 3, activated caspase 3. When I set up the assay, I provided the substrate in the form of a DEVD peptide. So these are basically the 4 amino acid sequence that caspase recognizes and cleaves next to. This DEVD peptide was bound to a small colorimetric molecule that binds to DNA when cleaved off. So activated caspase 3 in each one of our samples chewed up or cut up this particular linker and release the DNA tag to go in and bind to the cell DNA, and then that is the color that we look at, the colorimetric change in response we look at by spectrophotometric analysis. Now, if we were to load equal amount of protein in all our samples, we could have just looked at the OD or the absorbance value from the spectrophotometer just by itself and looked at how much activity was present. The higher the activity, the more apoptosis was happening. However, in our case, we had loaded equal volume and not equal amount of protein. So that is the reason that we had to do the calculation after the fact to quantify our proteins and to look at how much protein was present in each one of our samples so that we could do a relative caspase activity instead. We could have looked at that either through that or through a full change in activity compared to control. In our case, we made our untreated or the controlled sample as one and then looked at the activity of all the others in relation to it. We can also look at this stuff through DNA as well. This is what we would have done in lab this week and next if we had gone back to lab. However, in our case, we will be looking through mock data and some example gels to get the results. So for DNA fragmentation, you can do two different ways. We can extract the DNA and we can run it on a small agarose gel that is placed on top of a microscope slide. This is called a COMET assay. In this case, we label the DNA with a fluorescent marker so we can watch it move from in an electric field from negative to positive side. The DNA moves from the negative to positive side because the DNA is negatively charged due to the phosphate groups. If the DNA is intact and perfectly good quality, you will see nice concrete circles, very tight circles. If the DNA has started to lose its integrity because caspases are breaking it down, you will start to get a smear of some sort. The more smearing you see, the more DNA is fragmented and the higher apoptosis is present in that particular sample. This is called the COMET assay because it looks like a comet when there is fragmentation there. You can run it in a more clear fashion on a larger gel format where we run the electrophoresis in a big horizontal gel frame, and we look at it again by binding a dye to the DNA. We can do ethylium bromide or some other DNA binding dye to visualize the DNA. A nice clean band is going to indicate unfragmented, good quality DNA, and the more smearing you see, the more apoptosis is going to be present in each one of your samples. And here are two examples of samples that are undergoing various amount of apoptosis. Also, you can look at the intensity of the band to look at how much DNA is present. The more DNA is there, the more bright your bands are going to be. So you could look at that to kind of, do a qualitative analysis of, okay, it looks like there's a lot more DNA in my one treatment compared to the other. Okay, so that is all. This gives you a good, a little bit of background information on apoptosis. There is this video that I have available on there as well that you can watch regarding
One
apoptosis.
way that the development of cancer is prevented is via the death of defective cells. A cell that becomes mutated or damaged will first attempt to repair the damage. If that is not possible, the cells will commit the cellular version of suicide. An orderly process called apoptosis leads to the breakdown of key cell parts and the death of the cell. Cancer cells lose this critical capability and will continue to divide. This can lead to the accumulation of cells that can become more and more abnormal.
Okay, so I will leave you with that, and we are going to be meeting in class this week to discuss DNA extraction and analysis more in detail. Please make sure to go through the virtual labs before you come to class this week. Okay, take care.